Hello everyone, this will be the final lecture on blood and we'll be talking about hemostasis. Hemo means blood while stasis means stop. So hemostasis is the mechanism to stop bleeding. By the end of this lecture, you should be familiar with the structure and function of platelet cells which play an important role in blood clotting, the basic concept of hemostasis and how does a platelet plug is formed in a series of complicated intrinsic and extrinsic coagulation pathways. You also learn about the anticoagulant factors, which is part of our natural system to control blood clotting. And you should understand the process of fibrinolysis, which is the enzymatic breakdown of our blood clots, and several diseases and disorders that are associated with hemostasis. Of all the components in the blood, platelets are essential to hemostasis. So platelets are not whole cells, but rather they are fragments of cells budding from the megakaryocytes that are originated from the bone marrow cells. So under a light microscope, platelets are seen as these purple or bluish dots. They have a diameter of roughly 1/5 of the red blood cell. Now over here you can see that the megakaryocyte or the mothership for platelets is stained in purple or bluish by the Gemsa stain. So the megakaryocyte itself is much larger than the red blood cells and is only having a single nucleus. As a result, each platelet cell does not contain a nucleus because they are just derived from the cytoplasm of these megakaryocytes. Again, platelets are fragments of cells that lack nuclei. In other words, they are anucleated. However, they are equipped with organelles and enzyme systems that are required to generate energy and to synthesize secretory products. So they are able to contract themselves as well. So if you look at this diagram in more details, you could see clearly the presence of mitochondria, which is for energy production. And you can also see some Golgi apparatus or Golgi body to produce the transport and secretory vesicles. It also has granules to store all the materials and chemicals, and some cytoskeleton such as microtubules, microfilaments, and so on for contractile abilities, and they are all important in the function of hemostasis, which you will see why later. For a healthy person, your platelet count should be in the range of 150,000 up to 450,000 cells per ml of blood. Higher than this value, it is called thrombocytosis, while lower than this value, it is called thrombocytopenia. So this value can change depending on your health conditions, especially when you have something like viral infection or even chemotherapy that can affect the function of your bone marrow cells because all platelets are produced in your bone marrow. So during fever or dengue fever, a blood transfusion might be needed when the platelet count is lower than 10,000 cells per ml because that can cause internal bleeding. Okay, hemostasis is defined as the arrest of bleeding from a broken blood vessel. In other words, it is the stopping of a hemorrhage. But for bleeding to happen in the first place, there are two conditions must be met. Number one, of course, you will need a break or a cut in your blood vessel. Or number two, the pressure inside the vessel must be larger than that from the outside, so that blood will be forced out from the vessel. So this point number two actually forms the basis of first aid with bleeding, because we have to apply pressure from the outside to stop bleeding. So our body normally has the hemostatic mechanism in place to stop loss of blood through a small damaged capillaries, arterioles, or venues. So these are the three main strategies in place for hemostasis. The first strategy is known as vascular spasm or blood vessel constriction. So this happens only with the arteries, not the veins. I'll explain why in a while. So logically, with a smaller diameter with your arteries, you restrict the loss of blood. While for the second strategy, you have the formation of the platelet plug, which is formed by the aggregation of platelet cells. And the third strategy is known as blood coagulation, which is a strategy that involves a cascade of protein interactions to form the fibrin clots, and it is the ultimate step our body can offer to stop bleeding. We'll first look at the vascular spasm in more details. So when a cut happens, the smooth muscle surrounding the damaged vessel immediately constrict to slow down the loss of blood. So I mentioned just now that vascular spasm only happens in the arteries. That's because this mechanism is made possible by the presence of the smooth muscles. So arteries have lots of smooth muscle while veins have very much less. So how is this mechanism initiated? Well, it is initiated by chemicals known as vasoconstrictors that are released by the damaged cells and platelets such as serotonin and the ADP. When vascular spasm happens, the inner surfaces of the blood vessels are pushed together by the spasm of smooth muscles. You can imagine as this happens, the cut is partially closed up. And because platelets also secretes other chemicals, which makes them sticky and adhere to one another, they also add into the stop of bleeding. But bear in mind, vasoconstriction is only a temporal measure because the cut is not healed. But it does help to reduce blood loss and allow more time for repair and recovery to happen. So vasoconstriction is not always effective, as depends on which blood vessel is cut and how the blood vessel is cut. Therefore, we will need a second step, which is the formation of the platelet plug. So when platelets encounter the area of ruptured vessels, they will adhere to the exposed connective tissue, which is collagen, near the site of injury. And this structure of the endothelial blood vessel is known as tunica intima. So with this binding of platelets onto the collagen, that activates the platelets from smooth platelets into the spiky and sticky platelets. And it means they are now ready to clump together, forming the platelet plug. So this step of platelets aggregation is also assisted by a glycoprotein known as the von Willebrand's factor. So it is a plasma protein secreted by both the endothelial cell and the platelet. And what it does is to stabilize the growing platelet plug. So as more platelets are bound to the collagen through the help of this factor, they will release more factors or chemical factors such as ADP, serotonin, and thromboxane A2 stored within their granules. So the function of ADP is to add even more platelet cells, while the function of serotonin is to maintain vasoconstriction, and for thromboxane A2, it is to activate even more platelet cells for hemostasis. Eventually, the build-up of the platelet plug blocks the site of injury. But how do we know if the platelet plug is formed at the correct site? Well, the answer is our vessel, our normal, healthy blood vessel lining will release a chemical known as prostacyclin, and that inhibits the formation of the platelet plug because it inhibits platelet aggregation. It's only with the damaged blood vessel that do not produce prostacyclin, only then the formation of the platelet plug is possible. And aside from sealing the break, platelet plugs do have three other functions. Number one, platelets can contract and compact themselves to strengthen the structure of the plug. Remember I told you about platelets have a whole range of cytoskeleton to perform this function. Number two, the chemicals released from the platelets will cause further vasoconstriction, such as thromboxane A2, ADP, and so on. Number three, the plug also releases other chemicals which plays important role in blood coagulation that we'll describe later. Okay, here's a quick recap. When the blood vessel is damaged, that exposes the surface collagen. So it's the binding of the collagen with the platelet that activates the platelet cells, turning them from the smooth cells into the sticky and spiky cells. And not only that, the activated platelets will release some platelet factors, which are chemicals like ADP and serotonin, that could attract even more platelet cells. And with that, the platelet plug grows even larger. Now, on the other hand, when the endothelium is intact, this endothelial layer is going to secrete one chemical, which is prostacyclin, that prevents platelet plug from forming. So I think the key message here is that It is through the exposure of platelets onto the collagen that triggers platelet plaque formation. In your PowerPoint slide, I have attached a video which I think will be helpful to your understanding, so please watch the video. Moving on to the third stage, you have blood clotting or blood coagulation. Basically, it means the transformation of blood from its liquid state to the solid state or thrombus. So blood clot is also known as thrombus. Remember, you already have the platelet plug in place. So now the blood clot is going to form on top of this platelet plug, strengthening, reinforcing the structure. And this process is incredibly complicated. But ultimately, it involves the conversion of the soluble fibrinogen, which is a plasma protein, into the insoluble fibrin. And that fibrin structure could entrap the red blood cells by the mesh of fibrin fibers, as you can see in this picture. Okay, now let's look at the blood clotting pathway. We can say that blood clotting is a series of complicated cascade of reactions that involves a series of plasma proteins or blood clotting factors. In fact, there are 12 blood clotting factors altogether. And let's look at the last step of the cascade first. So in our blood, we have the soluble plasma protein, fibrinogen. Then with the help from thrombin, which is a protease, the fibrinogen is converted into the insoluble fibrin monomers. But eventually, these fibrin monomers will be assembled into the fibrin polymer with the presence of factor number 8 and the calcium ions. So there's an A over here, which means it is an activated form of the factor number 8. And this activation is done earlier by the glycosylation of this protein through a series of reactions. So in general, thrombin converts soluble fibrinogen into the insoluble fibrin monomers. Then these monomers are stabilized by polymerization, and it is catalyzed by factor number 8 that is activated, which requires calcium 2 positive, because calcium 2 positive will be the cofactor for this protein or factor number 8. So there are two separate pathways that can stimulate the activity of thrombin. Again, what is thrombin? It is the enzyme that converts the soluble fibrinogen into the insoluble fibrin monomers. So depending on the source of stimulus, if the damage is internal or within the blood vessel, the initial stimulus is collagen exposure, because collagen is just beneath the endothelium wall. So this pathway is called intrinsic pathway because all factors required in blood clotting are already present inside your blood. So even if you store your blood in a glassware, it will still clot, as it has all things, all factors needed for blood clotting to occur. If the damage is outside of the blood vessel, the damaged cells will secrete a tissue factor number 3, or better known as tissue factor, and this factor 3 will trigger a series of reactions that leads to blood coagulation. It is called the extrinsic pathway because the initiating factor is lying outside of the endothelial cell lining. Here is an overview to the blood clotting pathways. You can see that both the extrinsic and the intrinsic pathways lead to a common pathway in the center. So extrinsic pathway happens when there is a damage to tissue outside of the vessel. And the damaged tissues then release the tissue thromboplastin, which is also known as your tissue factor or factor number 3, that switch on the common pathway. Now the intrinsic pathway, on the other hand, happens when there is an internal damage to the blood vessel, such as when you're getting a bruise. So this exposes collagen. So it is the interaction of the blood cells with the collagen that triggers a series of reactions that finally switch on the common pathways. So for both pathways, their end product will convert the inactive factor 10 into the activated factor 10, which is an enzyme known as thrombokinase. So what does kinase do? They phosphorylates. So it phosphorylates prothrombin into thrombin. So as mentioned earlier, thrombin is the enzyme protease that will convert fibrinogen into fibrin. And with that, you will get blood clot formation. And here you have the extended diagram. Again, you can see that how both intrinsic pathway and extrinsic pathway lead to the common pathway. And the intrinsic pathway is activated or initiated by the contact of the blood cells with collagen. Then it activates a series of cascades with the presence of calcium ions. And eventually, the activated factor 9 will form a complex with factor number 8. And this complex then initiates the common pathway. So this whole process of intrinsic pathway takes a few minutes to happen as it involves many components and processes. Now, on the other hand, the extrinsic pathway will happen much faster because it has much few elements. So this pathway is initiated by factor number 3 when you have an external cut or an external damage to the blood vessel. So factor number 3 is also known as thromboplastin or the tissue factor. It then activates factor number 7 and combine it with factor number 3 to form a complex. So this complex in turn activates the common pathway. So in order to accelerate the whole process of the blood clotting, you have got two positive feedback reactions. It's either thrombin that can be fed straight into the intrinsic pathway, or the activated factor number 10 that can stimulate the extrinsic pathway. So this makes blood clotting a very fast and rapid process. And when the prothrombin is converted into thrombin, this enzyme can catalyze the conversion of fibrinogen into fibrin, which is the monomers that is insoluble. And with the help from activated factor number 8, you can form the polymer of fibrin that can cross-link and to form a mesh structure that can trap red blood cells. So that is your blood clot or blood coagulation. And here is another video that I highly recommend you to watch.